friends, welcome to another Sci video. In this one we're going to be looking at homeostasis and the nervous system as an introduction to the rest of the homeostasis topic. We're going to look at what the nervous system is after an introduction to homeostasis. We're going to look at how to do the reflexes required practical, the structure of the brain, the structure of the eye, and how the eye accommodates for long and short vision. So this topic is part of a larger topic of homeostasis. We're just covering 3 small sections today. There are other bits which will be in other videos. You can check out temperature control, there's hormone control, blood glucose levels, and then if you're biology, we've got more hormones involved in maintaining water and nitrogen balance. We've got hormones involved in reproduction, the role of adrenaline and thyroxine, and then required practical and plant hormones in biology too. But there are 2 other topics in this paper, ecology and inheritance of variation. Check out those videos as well. The first key question is, what is homeostasis? It is really quite simply the regulation of internal conditions. That can be for a cell or it can be for a whole organism, and it's about maintaining the optimum conditions. So this might be temperature, it might be pH, it might be the levels of sugar that are around. Ultimately, it's about trying to make sure that enzymes work as quickly as they can or as fast as they should to maintain cell function. And that's even for large organisms like us. If we don't have our individual cells working properly, then we don't work properly either. So we're going to look throughout the rest of this topic at some different types of homeostasis, blood glucose levels, temperature, water levels, to name a few, as well as how systems are controlled in plants. But the principle behind homeostasis is that receptors will pick up on what we call a stimulus, which is a change in the environment. They will detect this stimulus. So in the case of an eye, it is light. That receptor will pass information to what we call a coordination center, and that coordination center will decide what to do. So it might be the brain, it might be the spinal cord, and that will decide then which effector in the body needs to work. And these effectors can be one of 2 things. They are either a muscle or they are a gland. That's the only type of effector that we have in our body. So no matter the input, no matter the decision as the output, it can only be a muscle or a gland that works. A muscle can contract, that's all it can do, and a gland will secrete hormones or other chemicals like enzymes, things like that too. But the gland will secrete things, the muscles will do contractions, they will physically contract. We're going to look in the rest of this topic at 2 main systems in our body that control homeostasis. One is the nervous system, which is what we're going to focus on here. The other is the endocrine system, which is the hormone system. But the nervous system is just a collection of neurons, which are just cells, they're specialized cells. You probably saw them early in biology GCSE. You can look at the review of the cells topic and we cover specialized cells there. And these cells carry electrical impulses. They're quite strange because they can be very long cells. Cells are obviously really small, but these neurons, these nerve cells, can be very long. And in humans, we use the nervous system to react to our surroundings and to coordinate behavior. So we are responding by receiving information, deciding upon how to act, and then causing a response. And this here is a focus on reflex. Reflexes are really nice, simple behaviors. They are found in pretty much all organisms that have any behavior because they are the simplest type of behavior that we can find. And so the way that our reflexes, which we have reflexes like a pain response. We will flinch away from pain. That's a reflex. It's an automatic response, and reflexes are all about encouraging survival. That's what it is there for. It's been evolved to help us to survive. So we have different features in our body, different structures in our body that help us to have a reflex. So first we said we have a stimulus. The stimulus is an environmental feature. It's not a physical part of our body. The stimulus is a change in the environment. So that change in the environment is detected by a receptor. That could be a cell that perhaps detects changes in heat. It could be a cell that detects change in pressure, but that then passes information to a sensory neuron. So in the example here of little Timmy who's put his finger in a Bunsen burner flame, as some of your peers may well think is a good idea, he has here temperature receptors. in its fingertip. So the temperature receptors detect that there is a temperature change, and it sends this information along a neuron, which is all the way connected to here, which is the spinal cord. So this is a cross section of a vertebrae, and this will basically carry along just one whole nerve. So we're saying about nerve cells being really long. This is one nerve that's going all the way from the fingertip all the way to the spine, to this spinal cord. And so that's the sensory neuron, and it's easy to remember that because it's sensing, it's sending the information of what was sensed to the coordination center, which in this case is the spinal cord. Now it can be the spinal cord, it can be the brain, but for a reflex we don't want to waste time sending information up to the brain. We want to get it to the spinal cord and back out again. So for reflexes we have a really nice pathway. It goes from the sensory neuron to something in the coordination center called a relay neuron, which relays the message. I've got a mistake here, shocking. It relays the message via a motor neuron to an effector. So the relay neuron is just a bridge. It's this little bridge here. It's just connecting what is being sensed to what needs to happen. So there will be a few of these connections, and this in this case is nice and simple because it relays the message straight over. These are always existing, these connections. They're not made just for in this instance. It already exists. You have this response, this reflex already existing in your body. So change in temperature pings a message to your spinal cord, sends it across a relay neuron, sends it down a motor neuron, which here in this case is connected to a muscle. So the effector in this case is a muscle. We said it could be a muscle or a gland. For reflexes, you do have glands linked in too. So for this temperature response, if it is a high enough response, it might be that you get some adrenaline being released as a result of the reflex that's been triggered, because the adrenaline gland has been triggered to release adrenaline. The adrenal gland would do that. So in this case, a nice simple loop. We've got the receptor is in the finger. It sends information along the sensory neuron, which then get bridged to the relay neuron, sending it back to the motor neuron, which runs then to the effector and tells the effector what to do. The motor neurons are always determining what the outcome is going to be, the effector. And the effector causes something called a response, which in this case would be to move the finger away from the flame, for example. So lovely and simple. Stimulus picked up by the receptor sends the information along a sensory neuron, which bypasses the brain, going straight across a relay neuron, which sends a message along the motor neuron, which goes to the effector, and the effector causes a response. And what I tend to try and do is put these into little letters. You've got the stimulus, which is picked up by the receptor, which sends it across the sensory neuron to the relay neuron, which is then going to go along the motor neuron to an effector, which causes a response. More. Now we've got a lovely required practical here about reflexes, and the specification just has this title of investigating the effect of a factor on human reaction time. So the factor is what we're changing. That's our independent variable. So that might be something like caffeine. It might be exercise. It could be, I don't know, whether there's a distraction or not. But for most of these, you're going to have a control group and a group that has that variable. So you'd have a group which has no caffeine and a group then that has caffeine, and they both do the test. They do the test multiple times to get multiple repeats, and then you can take an average, a mean for those. The dependent variable is going to be this human reaction time. That's the dependent variable. And this practical actually comes up in physics as well, because when you're looking at reaction times for stopping cars, this is a nice way to link it in too. So the dependent variable is reaction time. But the typical method that you will have probably done is using a ruler and dropping it. So you have pupil number 1 holds the ruler, and you're dropping it in between someone else's fingers, pupil number 2, and they have to try and grab it as quickly as they can. So you don't tell them that you're going to drop it. It drops at a random time, and that person has to grab it. And how quickly they pick up or grab this ruler tells you how quick their reaction time. So they might grab it at this point 10 centimeters perhaps, and so they've got a measured time or distance, sorry, of 10 centimeters. They might do it again and get 12 centimeters. They might do it again and get 8 centimeters. These are pretty good reaction times. But they've done it 3 times. You take an average, you get an average of 10 centimeters. Now, our dependent variable here was reaction time. This is a reaction distance, it's not a time. So we need to convert it into reaction time. And we can do that by knowing the speed at which it's going, which is going to be always given to you in an exam question. It's based on the acceleration due to gravity, which is 9.81 meters per second. So with that, we can just use a constant value and you'd be given something to use to calculate that. But as a result, you would get a reaction time and you could then compare them. You could do a direct comparison, just distance. You could plot a graph of whatever variable is that you've been changing here. So this is the independent variable in this case. And your dependent variable would be reaction distance. Obviously, the slower the reaction, the greater the distance would be. And it might be that perhaps you've got a group that have had caffeine and a group that have not had caffeine. No calf. And you're looking for a difference, significant difference between the two. It's a lovely practical, but there are obviously lots of ways that it could be improved. You might be showing little nuance signs that perhaps you're about to drop the ruler. You're trying to help your mate and you give them a little nudge, or perhaps this ruler actually is sat a little bit further down in the person's hand. So it's not very accurate. And perhaps a better way of working with it accurately would be doing a reaction test on a computer where you see something flash up and you have to click a button. That would give a digital output at a time which would probably have a much higher resolution. And with a much higher resolution, your value would be much closer to the true value, so it would be a much better accuracy. And if you have a higher resolution, higher accuracy, it increases the validity of the conclusion. Validity of the conclusion is how trustworthy it is. And so with these things being done well, it would be more trustworthy. If you're doing separate sciences, you have 2 structures of the body that you need to know about, which are the brain and the eye. So we're going to talk about the brain here. And the brain is a phenomenal collection of millions, in fact billions of interconnected neurons. The number of synapses and connections between these neurons ends up going into the trillions, with each region of the brain having different roles, which is really cool. And we've only learned really kind of the surface of what the brain can do at this stage, because we've only really had methods of being able to find out how it works for the last 60 or so years. And a lot of the knowledge that we have of the brain comes from electrical stimulation and MRI scans. And before we had these techniques of being able to stimulate the brain or actually investigate it non-invasively, we had to use historical brain damage as a record. You might have heard of Phineas Gage in lessons. He's a bit of a legend in learning how the brain works. He had a pole that went through this part of his brain and his personality changed. So we learned that this part of the brain is in charge of controlling personality. But it is a very delicate and complex organ. If you've ever actually seen a dissection of a brain, perhaps you've done one in class, as soon as you remove it from the skull, it just kind of turns to mush. It's a bit gross, but the brain is phenomenally complicated, but so fragile. And so whenever we're trying to treat it or investigate it, it becomes really difficult. But there are 3 structures that you need to know within the brain. We've got the cerebral cortex, which is this main orange part. This is the most advanced part of our brain. We've got the cerebellum, which is a kind of legacy part of the brain. The lower part of the brain tends to be like the more basic part of the brain. It's the part that perhaps evolved first, and the cerebral cortex is what we've evolved and it's most complicated, and the medulla is right here at the bottom. It's part of the brain stem, and we need to know their functions. Now, the cerebral cortex is in charge of most of our intelligence. It's in charge of personality and language, a lot of processing of visual information, problem solving, conscious thought, high-level functions. That's all controlled in that cerebrum, or we call it the cerebral cortex in the GCSE. The cerebellum, this bit which is kind of lower back part of your brain, so kind of right at the top of your neck, that is in charge of balance, coordination, and muscular activity. But the kind of basic stuff, the automatic control of that sort of thing, you don't really use that part to do thinking of conscious movement, voluntary movement. And the medulla is the part that controls things like your breathing rate and your heart rate. So this again is really basic behavior. We don't have to think about that so much. You can think about your breathing, and now you're thinking about your breathing. But it's really basic, and basic organisms, basic mammals will have the medulla and the cerebellum, but they won't necessarily have a developed cerebral cortex like we have. Cerebellum was coordination, sorry. The only other part that we need to worry about a little bit later in the topic is this bit here, which is the pituitary gland, and the hypothalamus, which is in the core of the brain too. And these two are very important in homeostasis, but we don't need to know about their structures here at this point. With the eye structure, we're going to look firstly at their jobs, because these are important. So we should obviously know that your eyes detect light. They receive the light stimulus and they send that to the brain. But they also have two other jobs to help it to receive light stimulus. And that's different parts of the structure of the eye help it to do this. And one is accommodation, which is helping it to focus on near or distant objects. And that's the lens that has a job here. The lens is there to focus on near and distant objects as you adjust what you're trying to look at. And the second job is the adaptation to dim light, so making sure that your pupil is restricted or opened depending on how much light that you have. So to firstly start looking at these keywords, so the retina is this whole back part of the eye where light is actually going to be hitting the eye. So it passes in through the lens and the pupil, and then it kind of refracts and goes to various parts of the eye where it's then picked up. There are cells that will absorb the light and send that light information to the brain. The optic nerve is the bit that's going to be picking up this information from the retina and then sending it off to the brain. So the optic nerve is this bit down here, and this optic nerve then runs deep into the brain when that visual information is processed. So it's not telling us what we see at this point, it's just literally saying this is what light is hitting our eyeball. Our brain has to then make sense of it. The sclera is the outer region around here, the yellow part. It's just the outer protective layer of the eye, whereas the cornea is here at the front of the eye. It has to be completely transparent so light can pass through it, which is why it's a different structure. So that's the green part. The iris, you should probably know. The iris is a part of the eye that you see when you talk about eye colour. It's this bit here. It's coloured, and it actually is a bunch of muscle which can contract and relax. In fact, there's two types of muscle which will contract to control the pupil size. And the pupil is this gap here between the iris muscles. We then have ciliary muscles, which will contract and relax to help shape the lens, and that's found here and here. And they will actually pull the lens or relax to let the lens change shape. And we then have suspensory ligaments, which are the bits that are helping the lens to suspend. And these will also have an important role in adjusting the shape of the lens. And then the lens, of course, is this bit right here. So the light will come in, hit the lens, and then will change shape depending on what you're looking at. So you probably already looked at the brain and the eye structures in perhaps year 7 and 8, but you probably haven't talked much about how the eye will accommodate for objects which are far or near. And so we have two options here. First is looking at how they focus at near objects, and the second is how they focus on not near, on far objects. So first up, we'll look at the near objects, and we've got two structures which are in play here. The first is ciliary muscles. That word is so annoying. Ciliary muscles. And we've got suspensory ligaments. So to focus on a near object, we have the ciliary muscles contract and the suspensory ligaments loosen. What this does is it causes the lens to get thicker and then refracts light more so that it's bringing light back onto the retina properly. If we're looking at a far object, the ciliary muscles will relax, the suspensory ligaments will be pulled tight as a result of them kind of being pulled outwards by the muscles relaxing, and the lens is then pulled thin and only slightly refracting the light rays instead. And as again, the aim is to bring the image that you're trying to see into focus and landing perfectly on the retina, and that retinal information will then be sent to the brain to process the image. Sorry about my daughter crying in the background. Now, if this isn't happening properly, we have two eye problems that can come from it, and these aren't problems as such. A lot of people have not got perfect eyesight, and the two types of eyesight problems that we have are called myopia or hyperopia. Myopia is short-sightedness, and hyperopia is long-sightedness, where the image is going too far beyond the retina. Hyper meaning kind of high, it's going beyond, going too far. And you can see the image here, so it should be looking like this one at the bottom, where the image is forming perfectly on the retina. And myopia, it's falling short, and in hyperopia, it's going too far, it's going beyond the retina. So it is still going to hit, the image obviously is still going to hit the retina in both cases. It's just going to be distorted because it's not aligning on the retina at the right point. And so the image is going to be out of focus, which is why it looks blurry. And we can treat these easily. Most people will just wear spectacles or contact lenses, but we can also have laser surgery to basically reshape the lens to how it should be in its natural state. And we can also give replacement lenses if the lens is not shaped properly. Perhaps it's got cataracts, which makes it go cloudy and even more blurry therefore, or perhaps even a synthetic lens can be put in instead. That is it for our quick look at the nervous system, reflexes, brain and the eye. If you've got any questions or queries, pop them in the comments below, and do like and subscribe for more videos in the future.